This is the basic muscle structure. We first start out with the fibril, which is the smallest structure within the muscle, and then move on to a muscle fiber. Fibrils make up a muscle fiber, which then bundle together to create a fascicle. This makes up the basic structure of a muscle. This is a neuromuscular junction. When a signal is sent to a nerve, it interacts with a muscle to cause a muscle contraction.
First, acetylcholine is released from the synaptic vesicle. The nerve impulse arrives at the synaptic end bulbs, stimulating the channels to open. Calcium ions flow inward through the channels to perform exocytosis. During this, the synaptic vesicles fuse with motor neurons on the motor end plate, allowing acetylcholine into the synaptic cleft and fuse across. The mitochondria produce ATP, which is needed in order for the synaptic vesicles to release acetylcholine into the synaptic cleft. Next, the acetylcholine binds to the acetylcholine receptor on the motor end plate, and now that the channel is open, sodium can flow across the membrane. The muscle action potential is produced. The flow of sodium makes the muscle more positively charged. The change in the membrane potential triggers a muscle action potential. Then the action potential propagates along the sarcolemma into the system of T-tubules. It causes the sarcoplasmic reticulum to release its stored calcium into the sarcoplasm, and the muscle fiber contracts. A sarcomere is the basic contractile structure of striated muscle. The I-band includes a Z-disc and extends from either side of a Z-disc to myofilaments. The Z-disc holds actin filaments in place. The A-band extends the length of the myosin myofilaments within a sarcomere. The H-zone is the center of each A-band, where the actin and myosin myofilaments do not overlap and only myosin filaments are present.
First, a muscle starts out relaxed. Then, to contract the muscle, the calcium released by the sarcoplasmic reticulum binds to the troponin. It causes the tropomyosin to shift and expose the binding site. Myosin head is then energized when ATP is hydrolyzed by ATPase and is then split into ADP and a phosphate ion. The myosin head then binds to actin via the binding site. A power stroke will then occur, pulling the actin filaments, and in order for the myosin head to detach, a molecule of ATP is bonded to the myosin head.
Lastly, the acetylcholine is terminated. The acetylcholine binding does not last long because it is broken down by acetylcholinesterase. Quickly, the enzyme is attached to collagen fibers in the extracellular matrix of the synaptic cleft.